I'll leave it this morning.
I feel like
We're all in the
I know what you like save it and then save it and then arguably I find that like probably the most it's like 0 or
Yeah, I can just send you an email, but that's the reason why I don't just don't want to give up my space. Awesome, I'll send it to you after class today. Thank you.
His first reaction too was he was like, wait, you said that? It was sure. I was like, yeah. Like, okay, well, yeah, eventually. Yeah. Okay, I was saying, that's great. Yeah, like, everyone was chatting.
But last year, like, so
How do you like it?
so the first time, I decided that
Yeah, they were really
but the ones that happened in the town, they were
I think it's like, no, this is like this.
and then the beach and the beach the time of my but when that because I never beautiful all your beach will like become pretty much
Half an hour.
I want to do something for the beach or oxygen.
No, well, there you go. You should have seen me trying to pull on it today. I would remember.
Yeah, that's
Oh,
yeah. So what we can do is to pick the healthy one right now. All the same ones. One or two.
okay, yeah.
That's so from what I think. They clean with fish really well, but it won't be sensitive.
They show you the picture.
Especially that when the girls and I always ask the teacher to do the training. I think it's good. So they were so beautiful. Colors of them. That's it.
That's your dose?
You feel other
And right there, we're doing that one.
I just wanted to point out, get started, someone asked whether, bad graphs are still produced today and whether this happened in our sciences. So I know Carl Broman's website has his top 10 worst graphs, so if you want some entertainment, have a look there. Most of the examples are from this millennium and from genetics.
In what, sorry?
And are generally from the field of genetics, which is the area Carl Broman work. Okay, so I'm gonna hope that you can see these graphs with these overheads with the blind missing. I'll see what I can do about that. Maybe we can hang something there. So anyway, an outline today, and I want to focus on really why we produce graphs, some ideas on principles of display, and then a quick review of the kinds of graphs that we use to achieve these principles and some ways we might fail to achieve these principles, and how maybe to resurrect some of the problems that one encounters. A topic that didn't come up in the discussion, and there wasn't a lot of time for me to raise it at the end, was what about tables? So we also use tables in our presentations, and the question arises, well, what are they for? And I'll try and argue that actually tables are pretty much the same thing that graphs are for. They're meant to show patterns in data, not just to store as many numbers as you possibly can. So, yeah, as was already stated by the presenters, our human eye and brain are great at detecting patterns, so use that to advantage in trying to communicate your science, not just to other people, but also to yourself. So when you're analyzing your own data and you're trying to get a sense of what patterns are in there or whether there are outliers and so on, try a whole bunch of things. You use graphs. I use graphs all the time. It's the first thing I do, long before I calculate a p-value. This is, again, the colors are going to make it hard to see the patterns, especially from the back. This is a colored version of a famous graph by Florence Nightingale. And in it, she presented this graph to British Parliament. And what it showed was visually the causes of death to British soldiers in the Crimean War. And the area of the pie represents the number of deaths. And the deaths by wounds is in yellow. The deaths in red are deaths by infectious disease. And other causes are in blue. And with this graph, she showed vividly that soldiers die mostly of disease, not from bullets. And she persuaded and really persuaded the British government that whoever controls soldiers' health will win the war. And so graphs can change history. Good graphs can change history. It's kind of a confusing graph. No one would do this same style, but the visual impression was profound. Now, often we won't get a chance with our graphs and our theses to change history, but actually, a little bit. I bet the kind of data that we collect about the world will be useful, and what you would like to do, given that this is your chance, is to try to communicate that as effectively as possible. And so, how do we do that? So, what I thought one of the things I would do is to look at some of the graphs that we use and some features of those graphs that work, and maybe some features of those graphs that don't work. And generally when we create a graph for displaying our data, there's generally two types of data or two types of results that we're focusing on. One is to show the frequency distribution of variables, and the other is to compare that frequency distribution between groups, associations between variables. I wanted also to resurrect this graph, and also to say that a lot of the ideas that sort of initiated the field of statistical graphics were in this book by Tufte, and indeed he, among others, have said that this is one of the world's best graphs, and one of the best graphs ever produced. I agree kind of with both sides of the comments that were raised, because I think it's too complicated, the graph. But I never saw a graph like this in my history book when I, was a student in high school. And better than text and better than tables of numbers, this just awed me. The change in the number of soldiers and their locations moving from west to east and back again. The scale of the deaths that occurred during this misadventure in many ways would be difficult to describe. You could describe it with very exciting and breathless prose, but not all of us are great at doing that. But this is something that we can do. And one thing that Edward Tufte says, he has sort of a general principle that I'll talk about, but one of his statements is that a graph is not just to show stuff, to communicate results. It's also to put ideas into the head of the viewer, including yourself when you're looking at those graphs. And when I looked at this, I had a lot more ideas about Napoleon than I did when I was in high school, lacking this graph, because I wanted to know what did they die of, And when did they occur and how long did they stay at particular locations and so on. So graphs are powerful in that way. So how do we achieve this in our own graphs? Again, Tufte was really only the beginning, but it is a field now, statistical graphics. And that's relatively new. It really comes with the advent of desktop computers, laptops, and I say advent, I feel really old when I say that because it probably predates you, but me, I used one of the first terminals when I was a graduate student. And computers made it easy to try drawing graphs, to try different ways of doing it, so that because you're not drawing them by hand, you have lots of opportunities to get it right. So Tufte says it more beautifully than I do. Graphical excellence is that which gives the viewer the greatest number of ideas in the shortest time with the least ink and the smallest space. My paraphrasing of that, and maybe this is easier to remember, when you look at a graph, your own or it's someone else's, you should go, oh, not, huh? And that's a pretty good dividing line, I think, between a graph that works and does not work. So think of that as kind of a test of your own graphs. And if you're not sure, show them to your fellow students in your office, and if they go, what? Okay, you've got a result already. Okay, and so there's all kinds of lists of things that we could emphasize, but I want to emphasize 4. And the first is the most important, and that is to show the data. The second is to make the patterns in the data easy to see. The third is, well, something that this reading was all about, and that is to make sure you represent magnitudes honestly. And then, minimize clutter, make sure everything's the right size and so on, everything's readable. I want to emphasize also magnitudes, because something that you see a lot of when you read papers is you'll see chi-square statistics, you'll see degrees of freedom, you'll see p-values, but sometimes you have to look around for the actual magnitudes of things, the differences between the groups, the size of the differences between the groups, which I'm sorry to say isn't indicated by a p-value. And graphs do that. They at least give you an idea of how big the differences are between the groups in a way that is more difficult for text. And this is one reason why I think that one of the most important things you can do in a graph is to actually show your data. So Tufte, above all else, show the data. And there's a number of reasons for this. A common format for presenting graphs on numerical data is to show a mean and maybe a standard error bar. If not both, then perhaps one of them. Antennas. But this kind of a graph, although it's popular, doesn't actually show the data. It just shows the mean, the standard error. And actually all that information is located here. The rest of this bar might be useful for sort of giving the visual impression of magnitude, but in doing so, it hides this. And this is what I mean by showing the data, the actual points. And this graph from a PLOS biology paper, which was trying to encourage biologists to change the paradigm from this type of graph to this type of graph. And one of the reasons that he shows is that many different patterns in data can produce exactly the same mean and standard error. And you're doing yourself a disservice and also your viewers if you can't show them how this pattern came about, what the actual data points are. It communicates immediately the number of data points you have, or at least the density of data that you have. But also it tells you about outliers, it tells you stuff. And it also allows to say, well, here's the difference between the groups, but the variation within groups is also big, so let's put that in context. And that's something that we don't get from this method. So, Weisberger et al. Are trying to change the way we behave. And here's a couple of examples, and I hope you can see it. This is from my book with Mike Whitlock, where we try also to encourage people to consider, the problem of trying to display data. This is hippocampal volume ratio for a number of different treatment groups. The actual details don't matter so much, but I've drawn a graph with these data in two formats, and one is the commonly used format of a bar graph, and with the means and the standard errors. The other, the alternative way that I've presented it, the same data, is that I've shown the data. Now, I haven't actually shown all of the data points, but I've shown sort of with the box plots where most of them lie, and the range. This is the interquartile. There's the median, and so on. And these are sort of outliers. If you've learned how to draw box plots, then you have some idea of how to visualize these. First time you ever see one, it might take a minute. But I guess the question is, which graph is more effective and why? And this graph really loses none of the information that's in this graph. And it doesn't interfere visually with my brain, anyway, in presenting this additional information. But it tells me a lot about the variability within groups and how that compares with the differences between the treatment groups in that measure. Also that the variability shrinks as you get closer to 100. The distribution becomes more skewed as you get there, and so on. So, to my eye, this is a much better graph because it provides more information using less ink. The second recommendation is that you make patterns in the data easy to see. Now, this window problem is going to make this even worse, so I'm not sure if it's going to work. So you'll have to maybe download the PDF file from the website for the course to see this properly. But what this graph appeared in a paper that I thought was really interesting. And what the paper really showed is that the highest productivity regions in Africa not only have more birds, they also have more human languages. There's a kind of an association between those two. It's not 100%. And what this graph aims to show with this axis is that, where it gets dark purple is where language richness is high, and where more green means species richness is high, and then this combination is where this color is where both are high, and this color is where neither is high. And I found that even though there's a lot of information, it's possible to extract it visually from this graph. I kept having to go back to this. Okay, now what is this color again? Whereas I think it might have been easier to think of a way of displaying them more efficiently. But it's not easy, actually. I mean, I think that graphs is like any kind of analysis of your data. You may not get it right the first time. But because it's often the first thing that readers' eyes go to, and certainly for producing a talk, that it's well worth thinking about and making your readers, your viewers, when they see it, go, oh, I see it. I see the pattern in the data. 3, draw graphical elements clearly, minimizing clutter. So maximize the data to ink ratio within reason. And that leaves a lot of latitude for creativity. The question came up in the discussion, is creativity bad? I mean, one answer to that question is, well, is this creative? I'm not sure. There's a lot of ink spilled on this one. It's meant to show that people are getting fatter. This is year, 53, 54, 55, 56. And the height of the bar is, what is it? Already. It's hard. I'd have to go back to the legend to remind myself exactly what graphical element is showing what particular result. And as someone once said, well, it might be different for conventional media versus scientific reports. And it's true, we don't need entertainment. We're already entertained, we're scientists. Like, what improve on that? But in the media, clearly, more attention is paid to the infotainment aspect of graphs. But and sure, there might be some value in that. You want readers to actually stop and even look at the graph, because their motivations are entirely different. But there are ways to do this well, and ways to do this poorly. So the fourth point, represent magnitudes honestly. And yeah, another quote from Tufte. And I pulled a paper from Nature that interested me. And what this paper did was to show that birds sleep, but that only half of their brain goes to sleep at a time, and they alternate. So this was done on ducks, and it was scoring slow wave sleep score, and LO stands for left eye open, RO for right eye open, and when both eyes are open, their slow wave sleep score is low. So 100 is it's a relative scale, and so 100 is kind of the baseline. Whereas when their left eye is closed and their right eye is open, their left hemisphere is awake. Their right hemisphere goes to sleep when their right eye is open. Is that right? Right hemisphere sleep score. Yeah, and the reverse is true when the left eye is open. So they close one eye at a time and put the opposite side to sleep. So birds do this. I think they can even do this while they're flying. This was ducks sitting on the water. Birds are cool, aren't they? But this graph also has a problem. First of all, it's got a blue background. Why? But the other is that you see how traditions can die hard. Remember the bar graph with the antenna that is commonly used to indicate the mean and the standard error. So it was so important that the graph conform to this tradition that they made it into a bar graph. But then they ran into the problem that the graph would be far too long, far too high, in order to reach the baseline of 0. So what they did is they just sort of removed a section of it here. But the result of this now is that the height of the bar doesn't represent the magnitude. So in one sense, they sent it down to 0 so that the bar would represent accurately the fact that it goes from 0 to whatever this point is. But the purpose of bars in general is to give you an idea of magnitude, and then that's now distorted as a consequence. It's not even clear it's necessary here because 100 is actually the baseline, not 0 for this sleep score. So again, yeah.
So if you run into this problem where, let's say, the differences are significant, but if you start from 0 and actually go to 100, then it's not really clear that there is any difference. Whereas your data shows that it's significant. How would you represent that? Now, my second question is if you use the same method as you showed in a few slides ago with the box plots, wouldn't you also have to get rid of the space from 0 to 75?
So I guess there's a couple answers to the question, but the first is your last point, which is that show the data. Don't use bar graphs, use box plots. And then at that point you have to think, well, what are you trying to show, and is a baseline important? And that's a very specific question, specific to the actual project and goals and so on. But if your goal is to show differences between groups, there's no need to have a 0 baseline. If your goal is to show how far away they are from 0, that's a different matter. In this particular case, it also happens that 100 is actually the baseline. And if you're going to use bars at all, they should be deviations from 100, because that's sort of the reference of sleep. Nobody ever has a slow wave sleep score of 0. It's actually a meaningless number in this case, except I guess when they're dead. But a lot of decisions are made, and I have tried 3 times. I'm not going to show you any of my efforts, but I've tried 3 times to redraw this in a more effective way. And you know what? It's not easy. And so that's one of the reasons why we start with graphs, because they're so important, but actually it takes time to get them right. So here are some graphs that we customarily use that do seem to achieve their goals. So a bar graph for category data is pretty common, and I think we'll do one in our workshop this week. Why not? So this graph shows it was a study of the mortality caused by tigers in Nepal. So tigers still kill people in Nepal, and this particular study was aiming to show what they die of when they go into the national park where tigers are present, what their activities were, the idea being that maybe we could mitigate this somewhat if we had some data on what they were doing at the time that they were killed by tigers, which is an awful prospect.
If I ever go there, I'm spending a lot of time in the bathroom.
So I think in India, a colleague of mine told me that when the national parks people walk through the park, they always drag a goat. Just remember that next time you go. So anyway, these are the different categories. It's not that essential, but a bar graph is a great technique for representing frequencies, numbers of observations falling into a category of fractions. So that's the circumstance in which a bar graph actually makes sense, and also that it must have a zero baseline in that case. You're using the height of the bar to actually inform the eye of the magnitudes. So bars have to have zero baselines or they're inaccurate. And for category data, there's usually space between bars. The other thing that arises is, in order to communicate this sort of most effectively, how do you arrange the order of the categories? And the convention, and I think the best way to do this, is to go from the most frequent to the least frequent, because there's no other natural category for this data set anyway. Sometimes, if your data is the fraction or the number of people with a disease in 3 categories, then less severe, severe, more severe, then there's a natural order, but for a lot of these cases there is no natural order. Which reminds me, if you were going to produce a bar graph which showed the frequency of use of all the letters of the alphabet in the English language, what would you do? Would you start with A, B, C? Or would you start with E, which is going to be the most frequent letter, and then all the other ones following? Let's have a show of hands. You've had enough time to think about it, which is perfect. So how many would go with the ABC order? And how many with the most frequent to least frequent, but out of order? That's what I would do too. Except for the song, there really is no natural order to the letters. Is there? So the point about bar graphs was raised also because, pie graph was raised in the presentation. And here's another one. Here's the same data in bar graph format. And there's a lot of discussion online about bar graphs versus pie charts or pie graphs. And there's some research has been done on this. So people have drawn graphs this way or that way, and then asked people to estimate the proportion of the observations that are in this category, say, or various metrics that you might extract, not absolute metrics, but relative metrics. What proportion of the observations are in the most frequent category? Do this for pie graphs, do this for bar graphs. In this particular case, it would actually be easier for the pie graph. So I didn't find a lot of studies, and they don't always end up in the same results, but in general, it's easier to get a visual impression that's accurate of relative magnitudes from a bar graph than a pie graph.
I mean, do you think it would make a difference if you included like percentages or numbers on the pie graph? Because right now you tell them the numbers and the other one you don't in the pie graph. So
right.
You
I mean, in this case here, there is an axis. There's a scale. I mean, you could put the numbers on there, and that's sometimes what people do. I don't like it because I think it distorts the top of the bar, and we're trying to get the magnitudes right.
No, I mean like if you cut off the scale and gave them.
And then actually wrote the. So give it a try. Again, it's one of those things where, does it work?
Well, it seems to me like one of the most common places that pie charts, or at least a variety of pie chart, is seen is like when you're displaying haplotypes.
Yeah.
So what do you think about displaying haplotypes in the form of a pie chart?
So I've never produced a map, with different locations and the haplotype frequencies. My feeling is that when there's only 2 categories, it's not bad, but things go rapidly downward as the number of genotypes, number of haplotypes at a location goes up. So again, it's one of those things where you have a computer, it's easy to do, try different ways, and then ask yourself which graph makes you go, oh. And again, to return to the original point is, what are you showing us a graph for? Like, what exactly are you trying to indicate, and then use that as your reference as to what graph succeeds and which does not. Histograms, you're all familiar, we'll do one in workshop this week. This is a species abundance plot. So these are the birds at some national monument in Arizona, I think, or Texas. And each data point is a species, and along the x-axis is how abundant they were in the surveys, and on the y-axis here is the number of bird species in each of those abundance categories. So such graphs have been used to show the common pattern that most species are rare and relatively few are common. But again, it's a histogram, and the difference between a bar graph and a histogram is that whereas a bar graph, the height of the graph is mostly what your eye responds to, and that's accentuated by the gap between them. In a histogram, it's area. So there's sort of, you run these things together, and it's actually telling you almost the volume of data points in a particular region. And even not all histograms are of the same, the categories in histograms don't even have to be at the same, the intervals don't have to be at the same width, as long as the area is faithful to the volume. Now, associations between variables. Again, this is harder to do than just simple frequency distributions. So this is a kind of frequency distribution, but now we have kind of a 2x2 table to illustrate. So this is an experiment in which nests of great tits in Europe were sort of flagged, and eggs were removed from some of the nests, forcing the female to lay an extra egg. And then what they recorded was, at the end of the season, whether they were infected with malaria, whether malaria could be detected in these individual birds. And so what the graph is meant to show is the relationship between treatment, egg removal, and control. So that's that outer comparison. And then the response variable is the inner comparison, whether they had malaria or no malaria. Malaria and no malaria. Different colors are used consistently, as we've been told. And so then the question is, does this show the association, well, between treatment and control? And the observation is that whereas in the control treatment, most birds have no malaria, actually they're about equal frequency in the egg removal, indicating therefore that egg removal increased the incidence of malaria in the birds. But you usually have choices. This is another way in which sort of 2x2 frequency tables, contingency tables are represented using a mosaic plot. And this is almost a visual representation of the 2x2 contingency table, where now the area of the volume, the area of the bar, indicates the number of individuals in that category. And what this graph does is it dispenses with the actual counts and replaces them with the relative frequencies, and so they add up to 1 in both the treatment and control. Again, the convention is to have the treatment variable on the x-axis and the response on the y-axis, which is the convention for a scatter plot, for example. And if there was no treatment effect, then there would just be a straight line through here. That this bar would be down here. So it does tell you that this particular region has gone up, or is higher in the controls. It's gone down in the egg removal treatment. I'm curious to know, which did you find most effective in telling you that there was, or in visualizing the association between the treatment and the response to your eye? How many would go with the grouped bar graph? How many with the mosaic plot? That'd be interesting. The reason it's interesting, you have another.
I have a comment about the mosaic plot that like would be an issue is if you had two different ends, like for your treatment groups, like if you had more, because it just shows relative frequency, it doesn't show you how many of the control and how many of egg removal, and so that could like misrepresent your data in that sense.
Well, it's not misrepresenting in the sense that the volume, actually the width of these bars is proportional to the sample size.
Oh, okay.
Everything is proportional to the number of, so the way it's drawn is that these aren't of equal width, and so bar area is always proportional to the number of observations in that category. You don't know what the total number is though. And if that's truly an important thing, then the choice might be better for this one. And also I find that once you're up to 50 response categories, the mosaic plots start to do more poorly. So the general lesson is you have a computer, try them both, which works.
I think in this case they're kind of similar, but if you had more than 2 treatments, like 3 or 4 treatments, then that would get really confusing.
It can get visually confusing, and there's only one way to determine whether that's the case for your data. So we're seeing more and more mosaic plots, and we teach them in the undergraduate course here as well. When I started teaching this course, they were less familiar, and more people preferred this than this, but I detect a shift. Box plot. So I already showed you a box plot. But again, this is to show differences between groups or associations between variables. And what a box plot does is it shows the median. Sorry about that. If maybe more people could just kind of stand in the window, I would get less light shining off it. But the way a box plot is constructed is that's the median rather than the mean. And this is the first and third quartiles. And then this is most of the rest of the data, but then there's a criterion for what an outlier is. And I think an outlier is anything that's past 1.5 times this distance. But again, there's a large amount of data in this graph. And what you can see is that there's a positive relationship between this variable and this variable, but it tapers off. This is the clinical prediction to patients, terminally ill cancer patients, as to how long they have to live. And this is how long they actually live. And basically what it means is if the doctor tells you you have only 6 months to live, don't worry about it, because beyond that there's absolutely no utility. Okay, a strip chart is one way to replace the mean and standard error bars. In fact, this graph here, has you take the cake and eat it too, because it shows the data. And and we'll do this in R, and you can also jitter the data points so that they don't overlap like this. But then you also get to plot the mean and the standard error. I think maybe to do it in red was a mistake because tends to be confused as a data point, but those are the kinds of judgments you make. Here's sort of 3 ways in which you can compare differences between groups. So the data are hemoglobin concentrations in male subjects sampled from USA, sea level USA, and then high elevation Tibet, Ethiopia, and in the Andes. And this graph here is multiple histograms, where you just show separately for each of the groups a histogram. And if you're going to be doing that, you want them to be stacked vertically so that you can actually compare their locations. To have them side by side would be very, very difficult to actually do that. And then here's the box plot, and here's the strip chart. And I just showed this example quickly because, here's a case where there's so many data points in one of the categories that you really can't see the data with this style of graph. So maybe, again, try and see what works. Strip chart might not be the best idea then. Think of a box plot. A little bit of trouble with the box plot is it makes you think that there are way more outliers in the US, but actually there's just way more data points from that particular sample. So this one, I think, is perhaps the best in this case, but then again, only if they're vertically stacked. What this study, this is kind of a well-known study, but what they showed is that hemoglobin concentration in the Andes is higher than at sea level. But that's not the case in Ethiopia and Tibet, even though they also have enhanced capacities to take up oxygen. But the mechanism is different. Yeah, so the mechanism of high altitude adaptation to low oxygen and high altitude is actually different in different human populations in different island areas in the world, which is pretty cool. And scatter plots. So you've all seen scatter plots. And again, the explanatory variable here and the response variable there. And we've all produced scatter plots. We'll do one on Thursday, if you're good. And but the main purpose of scatter plot is very clearly to show the association between these two variables. To what extent does how colorful Father Guppy is? To what extent does that predict the attractiveness of his sons? We can see that there's a positive association. We get an idea of how strong that association is. But association is clearly what we're trying to depict here, not absolute attractiveness or ornamentation. And hence, there's really no need to have a zero baseline in a case like this. So again, the question of whether zero needs to be in your figure is always a case of just remember why you're doing this graph. And whether that is depicted honestly without the zero. If not, put it back. Paired data. So, the trouble with a conventional strip chart, say for two variables, two measurements, is that it kind of makes you think that every data point is independent, but often they represent measurements made on the same individual. So I'm back to this PLOS Biology paper on how good ideas for drawing graphs in biology. And here again is a case where a bar graph is used with the mean indicated by the top of the line and the antenna indicating a standard error. So not only does this fail to show the data, but it fails to show the relationship among the data. The fact that this is the same individual as this one. And so paired data can be depicted using a plot like this where the groups, they repeat measurements on the same individual, are connected by a line. Sometimes that's called an interaction plot. I think there's a command in R called interaction.plot that does that style of graph. Or, since the independent data point for paired data is the individual, and not the two measurements themselves, the alternative is to do a strip chart, in this case on the difference, because that's the independent data point. Does that make sense? And then what do you do if there's more than two categories? Well, there are ways of, so here are 4 flower measurements taken on a group of individual plants. This is some data set that circulates and is used a lot in showing methods in R. There's a website, R Graph Gallery, where I got this, where you can look at graphs that people have created in R, and some of them are truly wondrous. So group data like this is like paired data, same problem, except now you have 3 or 4 measurements on the same individual. So in order to prevent yourself from distorting the reality, which is that these are the same individuals measured multiple times, well, there are graphical ways of depicting that as well. And the result is that maybe different species have different relationships between those two variables, and in this graph that's indicated by color. How not to display category frequencies. We've all had a good time reading this paper, the reading for the week, where there's some truly amazing examples of distortion. But we do this too. Cell is, maybe the best journal in cell biology out there. This is 2004, relatively recent. And this is a category plot, which could be displayed with a bar graph or with a pie. And there's sort of two features of this graph that, to my eye, make it less effective. One is it's 3D. And what 3D does is it actually distorts the magnitudes, the areas of the bars, because these, this bit here includes all this, whereas that one doesn't. So immediately your eye is tricked, and the magnitudes are not represented accurately. And the second is, well, making it 3D, it's on its side, which again makes it harder to visualize the areas, which are already difficult enough in a pie graph to begin with. And there is the sense that the most useful data in this graph is not the graph itself, but the numbers that are around it, in which case this is just a waste of ink. A table is a beautiful way of providing those numbers. So I think maybe I got this from Carl Broman. A graph that's meaningful only with numbers added must be recognized as a failure. But even 2D pies, as we've seen, can fool. But again, in deciding whether this is a good graph or a bad graph, we have to ask ourselves, what was the point of this graph? What was it trying to do? Turns out, the aim of this graph was to show that changes in NO3 and SO4 between winter and summer, those concentrations changed with time of the year. And then you have to go, okay, which is SO3, which is NO2, and why are the other categories there? And then, and also that the change was consistent among geographic regions.
So I'm assuming that they put all of the other chemical composition, like the other chemicals in, because they felt like without having those, they're not
Yeah, they would
accurately
be
displayed.
cherry picking or something like that.
Yeah.
Yeah,
So,
absolutely.
what would you recommend in this case if you wanted to not cherry pick, but also just show this in a clearer way what they're actually trying to show?
So, that's your burden, not mine. Because you've hit upon the essential question. Okay, we can all agree that it's actually really hard to see the main pattern that the authors wanted you to see in this figure. So we know that surely we can do better. But exactly what constitutes better, you have to try. My next thing to do would be to do a line graph where I have the different seasons along the x-axis maybe, and then one for NO2 and one for SO4, NO3 and one for SO4, and that would allow you to include all these other categories that'd be on the bottom, so they would actually be separated to some extent from the higher numbers, which are the ones you're focusing on anyway. So I think there are ways of doing that, but I have to draw it to see whether it would just be a mess or not. Here's a graph, ratio data. So this is a summary of a meta-analysis in which the authors used biomass ratio to examine the effectiveness of marine protected areas. And so every marine, there were 32 marine protected areas, and the biomass of marine plants and animals inside the reserve was compared with that at a representative site outside the reserve. The ratio of those two is the data point for that particular reserve. And the values of that ratio are then illustrated here. And so if there's a value of 1 means they're the same. And so there's no difference overall between the inside and outside the reserve. And this was one of the early meta-analyses that seems to indicate that there's more biomass inside reserves than outside. That ratio is more than 1. But can you see an issue with this graph? It's not that easy.
I don't know what the bar is actually
actually like what the number of the bar is, because it goes from 0.5 to 1? Like, is that ratio 0.5? Is that what it's supposed to be?
It's somewhere between the values 0.5 and 1.
Okay,
So it's a
okay.
histogram. So these are sort of
Okay,
bins,
yeah,
and then you're counting the number of observations that follow them. It's not a bad graph, but ratios do have one sort of flaw, and that is if the ratio is greater than 1, it can vary from 1 to positive infinity. And if the ratio is less than 1, it can vary from 1 to 0. So the scales are not really comparable on the two sides of the sort of the null hypothesis point, which is this one here. And so, again, fairly subtle, but it's easy to trick the eye in a way that might make you think that there's more area on this side, which there clearly is in this case, so it's not truly a distortion. But so you have to think about these things. How would I sort of equalize the scales to the left and the right of 1? Well, one simple thing to do is take the log of the ratio, and then everything is, once again, on the same scale. Right, said that. Tables. Why do we produce tables? Well, one thing that tables can be used for, like the multi-pie chart graph that I showed you, is sometimes the temptation is just to use them to store all of your numbers. But if that's your purpose, I think that they should go into the appendix. Nobody wants to see your stored numbers. If you're going to show a table, you really should use that table to illustrate something, a pattern. A table is also a visual representation of data. It summarizes information, and the question is, how do tables work? And my feeling is that the same goals that work for graphs should also work for tables. Acknowledging the fact that they have different purposes. So here's the tiger mortality data. Again, here's the frequency table that accompanies it. And there's always the decision of what's better. What's a more effective way of showing the relationship between what people are doing at the time that they're killed at tigers, the actual numbers or a visual representation of those numbers in a bar graph. Again, to some extent that will depend on your goals, but in the same way that you start with the most common category and go to the more rare ones in the graph, you should do the same thing in a table, because really you can more easily compare them if they are ranked in some way, particularly if the categories themselves are not ranked. So that's a simple way in which a frequency table can accomplish some of the goals of a graph. The other feature of this frequency table is that the numbers are aligned this way that you want to compare. For reasons that are not clear to me, if I had written these numbers horizontally, they would be more difficult to compare than if they're one on top of the other. That's just the way it is. It's just the way our brains work. So there are ways to improve tables too. Here's an example. I like this one. So what this graph shows is the different king-queen, the different Spanish Habsburg kings and queens, the inbreeding coefficients of their progeny, and then various metrics on like survival of those progeny. And the idea of the table was that it's not a huge amount of data, but it was exploring the possibility that there was a relationship between inbreeding coefficients and the survival of princesses and princes. But this is a horrible table in my mind, because look at all the gaps in there. It's really breaking up the numbers. Secondly, the variables that we most want to compare are the ones on the right and this one on the left. Here's the sample size. Get that out of there. We don't want to compare F with sample size, and yet those are the variables that are adjacent in the table. Finally, I think they're in chronological order rather than in interesting order, making it harder to see what that association actually looks like. So you can redraw that table like this. Cut down the number of decimals so they can read the numbers faster. Order the kings and queens not chronologically but by F statistic. and then just ask, is there sort of a general tendency for the numbers in these other measures to be larger down at the bottom of the table than at the top of the table? So a table can also draw the eye to relationships that might be present or might not. Your purpose might actually be to show that the data indicate no relationship, but that's also a goal, if that's what the data show. So think of your tables as well as your graphs. Finally, the one last topic I wanted to mention was graphs that move. So it wasn't a topic that's raised yet, but more and more I'm seeing people publish papers where the graph is a little sort of moving. And these can be powerfully effective. I hope this will work. Let's see. This is from the R Graph Gallery. I've got the site, but it's not loading. Yeah, there we go. So this is a 3D graph, which are generally horrible. They're very, very difficult often to see. And what it does is, it's a movie in two senses. One is that it's turning, so you get to see the data from more than one perspective over the time that you're watching it. But then it also, it's a movie in the sense that the different groups are added as you go. In my mind, it's a question: is this good? Does this help? Think of your objectives before you waste a lot of time trying to construct a little bit a moving graph. I don't know if you've ever seen some of these other ones. I won't show you this one because there's a voiceover and everything else, but have you ever seen the video statistical graph called The Fallen of World War II? I highly recommend it. This is another movie graph, so it attempts to show data in time, year by year, but time is actually the third axis, so the graph changes as time goes on. It's a 2-minute movie, and the goal is to show the numbers, which, and so here's a map of, again, the power of this graph is enormous, but it's diluted a little bit by the lighting issue here. So Africa, Europe, Western Hemisphere, and each of these dots is a ship filled with slaves from Africa. And so what the graph shows you in time with a video is basically how many ships there were at different points in time during the period that there was a slave trade. And what it shows along the bottom here are the sort of the cumulative numbers. And so we're here down in, are the years. So we're here still. So if you've ever doubted that graphs can move people, here's one example. It's one of my favorite examples. Almost appalling. And I think this is, I mean, it's hard to represent in just text. Look at this. When I watched this movie, I kept thinking, were there any to Canada? Were there any to Canada? Were there ever any slave ships from Africa to Canada? And the answer is yes, but not from Africa. They came from the US.
Has anyone ever using the Gapminder software?
Yeah, that's another famous video. So this Swedish guy gave a TED talk on Gapminder and showed a game with a movie in real time. And he's standing in front of the screen talking, and his arms are waving, and he's with his hands he's moving the data points across the screen. Also, very impressive movie graph. So. If you ever wanted a visual impression of this horror, then here it is. So, before you all run, let me do one more thing. And that is the reading for next week. Okay, it's on the webpage. Information's there. I need 2 presenters for next Tuesday and 2 moderators. If anyone would like to do this, come forward and talk to me now, once we end. The second thing I wanted to point out is that there's an assignment on the webpage as well. So the idea of the assignment is this. Find a bad graph published by your supervisor. Analyze it. Why does it not succeed? Believe me, I'm going to hear from your supervisor, but don't let it worry you. And then I want you to improve it in R. So we've talked about principles, the goals of a good graph. Analyze this graph. Why did you choose it? Why is it a bad graph? What is it trying to do? Why does it fail? Then I want you to redraw in R, and if you must simulate some data, do so in order to do the improvement. And if you have any questions about that or need to make some judgments, just ask me. And then once you've drawn your improved graph, analyze it. Why does it work? And at the very end, I want to see your R code. So more details are at the website as to exactly what's expected.
Friday, I just had it.
Friday, so I have a teaching tutorial.
Consider application.
So I could stay for an hour.
So 2 works.
2 does not. Actually, I had
The real topic is experimental design. Okay, so